Hi, my name is Tejas Athavale, and I worked with Luai Jawad, Dr. Krisanu Bandapadya, and Dr. Elena Zhang on our project on the bioconjugation of gold nanoparticles. So the bioconjugation of gold nanoparticles has been a very hot topic in the past few years. Gold nanoparticles are being researched for their use of drug delivery to different cells in the body because of low levels of toxicity and ease of use. Gold nanoparticles are between 20 and 200 nanometers and are able to cross the blood-brain barrier and deliver useful medications to specific cells within the brain. Gold nanoparticles not only cross the blood-brain barrier easier than using drugs alone, but they also increase the half-life of the drugs, thereby decreasing the necessary dosage to achieve the same results. So some more background and our purpose. Proteins are synthesized from amino acids at the endoplasmic reticulum on ribosomes in the cell. Disturbances in calcium regulation, glucose deprivation, and viral infections can cause ER stress, where certain essential proteins become misfolded. Prolonged ER stress gone untreated can lead to cell death by apoptosis. ER stress can be combated using certain drugs like 4-phenylbutyric acid, 4-PBA, which acts as a selective inhibitor and chemical chaperone and stops protein misfolding and apoptosis. Our project seeks to make 20 nanometer gold nanoparticles in solution through the Turkovich method, which I'll explain in a little bit, to deliver 4,4-aminophenyl butyric acid, a derivative of 4-PBA, to targeted cells in the brain and to suppress the unfolded protein response, UPR. By suppressing the UPR in a more efficient manner, our particles can prevent neurodegenerative diseases such as Alzheimer's or Parkinson's. So here's our procedure for the initial synthesis of the gold nanoparticles all the way through to the final attachment of the drug to the particle. So in the first step, we want to synthesize our 20 to 30 nanometer gold nanoparticles. In the second step, we want to attach a compound with a carboxylic acid terminal group to the surface of the nanoparticles. The carboxylic acid terminal group will make it easier for when we go to attach the drug to the particle at the end. For our third step, using EDC and sulfo-NHS in a cross-linking reaction to form an amide bond between the carboxyl and the amine-terminated drug. EDC and sulfo-NHS are compounds that we will use in our solution when we are making the drug and the particle, which allow the bonds to form in an easier manner. So for the actual nanoparticle synthesis, this is the Turkovich method. In step 1, we will make a solution of chloroauric acid in millipore H2O. For step 2, we heated the solution under reflux to 100 degrees Celsius. Once the solution began to boil, we added a solution of trisodium citrate, which acted as our reducing agent. In step 4, we allowed the reaction to run for 15 minutes and noted any color changes. Finally, once the reaction was completed, we characterized the particles using dynamic light scattering, UV-Vis spectroscopy, and zeta potential analysis. The photo on the right shows our final solution once it had run through reflux. You can see the cherry wet red wine color, noting that we do have gold in our solution. So here's some more of our data and results. The photo on the left is a little bit different color because it's from an earlier trial when we were running experiments. It's the same chloroauric acid post-reflux with the sodium citrate. On the right, we have our UV-Vis spectroscopy graph of our 20 nanometer gold nanoparticles. The peak at wavelength 522 nanometers is indicative of gold in our solution and helped us knowing that our experiment was working. So some of our future work is modify, functionalize, and verify our particles. Through modification, we want to attach the carboxylic acid terminated compound to the nanoparticles to make it easier to attach the particle to the drug. The compound we're using is 11-mercapto-undecanoic acid, or MUA, due to the thiol group on one end having a high affinity for gold, allowing for stronger and easier bonding and attachment. Through functionalization, we will be using EDC and sulfo-NHS to attach 4,4-aminophenylbutyric acid. EDC will activate the OH group at the end of the MUA. Sulfo-NHS will stabilize and increase the efficacy of the reaction to form the peptide bond once we add in the drug. pH conditions will be optimized based on the MUA and the drug in solution. For verification, we will be using nuclear magnetic resonance and transmission electron microscopy to characterize the shapes of the final particles, making sure that we have obtained our desired product and that no conformational changes have occurred. So some more on our future work we're planning. We want to affix a fluorescent molecule to our particles. Attaching a fluorescent molecule to our particles will allow us to track them and ensure that they're going once they have been injected. We would also like to couple a ligand to help the particle cross the blood-brain barrier. We are expecting the particles to cross the blood-brain barrier by passive diffusion. However, if that is found to be too slow for our purposes, we will attach a ligand, such as transferrin, which has a receptor on the surface of the endothelial cells comprising the blood-brain barrier, to our particle to allow for receptor-mediated transcytosis, RMT, into the brain. RMT will significantly increase the uptake of our particles. Another possible option is coating the gold nanoparticles in insulin using EDC and sulfo-NHS, and using insulin receptors to cross the blood-brain barrier, same as transferrin, through RMT. But further research is needed for non-diabetic patients. Here are references that we have used so far throughout this project. Again, I would like to thank Luai Jawad, who has been helping me in the research lab making the particles and coming up with ideas. I would also like to thank Dr. Krisanu Bandapadhyay, who has been guiding us from the beginning and telling us the procedures that we need to work on to make the particles. Finally, I would like to thank Dr. Elena Zhang, who is the biology professor that we have been working with. She is the one that gave us the drug that we needed to attach and has been helping us tune our ideas and tune the particles in a way that will make them more efficient. Thank you.